In this tutorial, we'll show you how to use our online tool called TOAST to contribute information about Arabidopsis genes to TAIR. TOAST is used to capture experimental information about Arabidopsis genes from the literature. In TAIR, we capture gene function data and expression data from individual papers using terms from the gene ontology and plant ontologies. The main types of annotations we make in TAIR use gene ontologies to describe the molecular activity of a gene product, such as kinase activity, its biological role, such as signal transduction, and cellular localization, such as in the plasma membrane. We also use TOAST to capture experimental information about gene expression and protein localization within the whole plant using plant ontology terms, as well as capturing protein binding interactions. Having the experimental data captured in a structured way makes it easier to find and easier to manipulate computationally. It also makes your data more discoverable to the research community, and of course it contributes to making Arabidopsis the gold standard reference plant genome. To access TOAST, start from the TAIR homepage. Go to the section marked Submit and choose Online Submission for Authors. To use TOAST, click the button to fill in the online form. You'll need to be registered with TAIR to use TOAST and use your TAIR username and password to log in. Once you're logged in, you'll be presented with the form and asked to enter in a unique identifier for the paper that you're curating. You can use either the PubMed ID or the digital object identifier that was provided by the publisher. This is so all annotations can be linked to the publication that's the source of the data. Next, you'll add the unique AGI locus identifier for the published gene that you want to annotate. If you don't know the AGI code, you can perform a search in TAIR. If you've given the gene a different name, you can add the full name and symbolic name here. The annotation section is divided into different categories for each of the main types of annotations we capture. You don't have to enter information into all of the fields, only for those that have experimental data in the paper. To choose a term, start by entering in text, and the autocomplete function will show the ontology terms that match. For each ontology, only the terms from the relevant ontology will be shown. Choose the term that is the best match. If you can't identify an appropriate term, enter your own suggested term. Next, you'll select the type of experimental data from the paper that supports the annotation. Again, choose the best fit from the drop-down menu. Do this for as many types of annotations that you wish to make for that gene. If you need to make more than one annotation of any type, you can click on the plus sign in that category to add more data entry fields. For example, if you want to annotate more than one biological process. We also capture information about protein-protein physical interactions. If you have information in the paper about protein interacting partners, you can enter the name of the binding partner here. And again, provide the experimental method used to demonstrate the protein interaction. If there's any additional information about this gene that you'd like to share, you can enter comments in this last field. This might be a good place to add information that's not captured above, such as genetic interacting partners or DNA binding targets. If you're done adding data for this gene, you can submit your data now. Or if you have information about additional genes, such as a genetically interacting partner described in the paper, you can add fields to capture information about this second gene. Please be aware that you can't save your work in progress, so once you've completed one annotation, if you need to stop at any point, we recommend that you submit your annotations and come back and complete the rest later. When you're done adding all of your annotations, click on the Submit button at the bottom of the page. Once your annotations are submitted, they'll be reviewed by a curator. If we have any questions, comments, or changes, we'll contact you by email. Once your annotations are approved, they'll be shared with the community. We thank you for your support, and if you have any questions, please contact us at curator@arabidopsis.org.